Welcome to our exploration of mitosis, the fascinating process of cell division that keeps life going. In this video, we'll journey through the stages of mitotic cell division in both plants and animals, uncovering how our bodies grow, heal, and maintain themselves. We'll see how chromosomes play a crucial role and how the cell cycle orchestrates the entire process. Whether you're curious about how your skin repairs itself or how a single fertilized egg becomes a complex organism, mitosis is at the heart of it all. Let's dive in and discover the secrets behind cell division. Have you ever wondered why your skin heals after a scrape or how your body grows taller? The answer lies in cell division. Every second, millions of your cells die or get injured and they need to be replaced. Without this constant renewal, your body would shrink and eventually cease to function. Cell division is also essential for growth, allowing organisms to increase in size as their cells multiply. In single-celled organisms, division means reproduction. One cell becomes two, ensuring the continuation of life. Let's see why this process is so vital for all living things. It's a fundamental principle of biology. All cells come from pre-existing cells. There are no exceptions. Every one of the 100 trillion cells in your body can trace its lineage back to a single fertilized egg. This remarkable continuity ensures that life persists generation after generation. The process of cell division is the engine behind this continuity, allowing organisms to grow, repair, and reproduce. As we move forward, keep in mind that every cell you see today is the result of countless divisions stretching back through time. Cell division serves three main functions: replacement, growth, and reproduction. Replacement is crucial because millions of cells die or are injured every second. Without new cells, our bodies would shrink and fail. Growth happens as cells reproduce, increasing the total number of cells and allowing organisms to get bigger. In single-celled organisms like bacteria, cell division is reproduction. One cell splits to form two new organisms. These functions highlight why cell division is essential for life, from the smallest microbe to the largest animal. Cell division is a tightly regulated process, but it raises some intriguing questions. How do cells know when to divide? Why do some cells, like those in calluses, divide rapidly while others, like brain cells, rarely do? And why do certain cells, such as blood cells, constantly divide to keep up with the body's needs? Scientists continue to investigate these mysteries, seeking to understand the signals and mechanisms that control the timing and rate of cell division. These questions are at the forefront of biological research. Life of a cell is a continuous cycle, consisting of two main phases: dividing and not dividing. The dividing phase is known as mitosis, a relatively short but crucial part of the cell's life. A much longer phase, called interphase, is when the cell is not dividing but is busy growing, repairing, and preparing for the next division. Understanding the cell cycle helps us see how cells balance their time between these two essential activities, ensuring proper function and survival. Interphase is the longest phase in the cell cycle, where the cell grows, repairs itself, and prepares for division. During this time, the cell absorbs nutrients, repairs any damage, and duplicates its chromosomes in the nucleus. For human cells, this means going from 46 to 92 chromosomes, setting the stage for mitosis. Chromosomes during interphase appear as long, thin strands, often compared to spaghetti. This phase is all about getting ready for the big event, cell division. When a cell is ready to divide, it enters mitosis, the process where the mother cell splits into two identical daughter cells. The final step is cytokinesis, where the cell's cytoplasm divides, creating two separate cells. This ensures that each daughter cell receives an equal share of genetic material and cellular components. Mitosis and cytokinesis work together to maintain the continuity of life, allowing organisms to grow, heal, and reproduce. Mitosis is a step-by-step process with 5 distinct phases. Each phase plays a specific role in ensuring that the genetic material is accurately divided between the two new cells. We'll follow the journey of a human cell as it moves through these stages, starting with prophase and ending with telophase and cytokinesis. Understanding these phases helps us appreciate the precision and complexity of cell division. First phase of mitosis is prophase. Here, the long, thin chromosomes begin to shorten and thicken, making them visible under a microscope. The nuclear membrane dissolves, allowing the chromosomes to move freely within the cell. At this point, the cell contains 92 chromosomes as they were duplicated during interphase. Prophase sets the stage for the orderly distribution of genetic material. During metaphase, the double-stranded chromosomes line up in the middle of the cell, forming what's called the metaphase plate. Spindle fibers extend from structures called centrioles, attaching to the chromosomes and preparing to pull them apart. The cell still has 92 chromosomes at this stage. Metaphase ensures that each new cell will receive an identical set of chromosomes. Anaphase is the stage where each chromosome splits and the two halves are pulled to opposite poles of the cell. This movement ensures that each daughter cell will receive an identical set of genetic material. Now, 46 chromosomes move to each side and they are single-stranded. Anaphase is a critical step in maintaining genetic consistency from one generation of cells to the next. In telophase, new nuclear membranes form around each set of chromosomes at the cell's poles. The chromosomes begin to uncoil, returning to their less condensed state. This marks the near end of mitosis as the cell prepares to split into two. The formation of new nuclei ensures that each daughter cell will have its own complete set of genetic instructions. As telophase ends, cytokinesis begins. The cell membrane pinches in, eventually splitting the cell into two separate daughter cells, each with 46 chromosomes. This final step ensures that both new cells are identical to the original mother cell. Cytokinesis is the physical division that completes the process of mitosis, allowing life to continue seamlessly. Cytokinesis occurs differently in animal and plant cells. In animal cells, the cell membrane pinches inward, forming a cleavage furrow that eventually splits the cell. In plant cells, a cell plate forms between the two new nuclei, gradually developing into a new cell wall that separates the daughter cells. These differences reflect the unique structures and needs of animal and plant cells during division. In plant cells, cytokinesis involves the formation of a cell plate. This structure grows outward from the center of the cell, eventually fusing with the cell membrane and forming a new cell wall. This process ensures that each daughter cell is encased in its own protective wall, a necessity for plant structure and function. The cell plate is a key adaptation for plant life. In animal cells, cytokinesis is achieved by the cell membrane pinching inward, creating a cleavage furrow. This furrow deepens until the cell is pinched in two, resulting in two separate daughter cells. This flexible method is well suited to the soft, dynamic nature of animal cells, allowing for rapid and efficient division. Cell cycle is a continuous process, with each phase flowing smoothly into the next. After cytokinesis, the new daughter cells immediately enter interphase, beginning the cycle anew. This seamless transition ensures that cells are always ready to grow, repair, and divide as needed. The continuity of the cell cycle is essential for the maintenance and health of all living organisms. Let's revisit interphase, the phase where cells spend most of their time. Here they grow, carry out their normal functions, and prepare for the next round of division. Nutrient uptake, DNA replication, and repair all occur during this phase. Without a thorough interphase, cells wouldn't be ready for the precise steps of mitosis. It's the foundation for healthy cell division. During prophase, chromosomes condense and become visible, the nuclear envelope breaks down and spindle fibers begin to form. These changes are crucial for ensuring that the chromosomes can be accurately divided later on. The cell's internal architecture is reorganized to facilitate the upcoming steps of mitosis. Prophase is all about preparation and organization. In metaphase, chromosomes align at the cell's equator, attached to spindle fibers from opposite poles. This alignment ensures that each daughter cell will receive an identical set of chromosomes. The cell checks that all chromosomes are properly attached before proceeding, a critical quality control step in mitosis. Anaphase is marked by the separation of sister chromatids, which are pulled toward opposite poles of the cell by the spindle fibers. This ensures that each new cell will have the correct number of chromosomes. The movement is rapid and precise, highlighting the cell's remarkable organizational abilities. Telophase reverses many of the changes seen in prophase. New nuclear envelopes form around the separated chromosome sets, and the chromosomes begin to decondense. The cell is now ready to complete division through cytokinesis. Telophase ensures that each daughter cell will have its own nucleus and complete set of genetic material. Cytokinesis is the final step of cell division, physically separating the two daughter cells. In animals, this involves the cell membrane pinching in. In plants, a new cell wall forms. This step is essential for creating two independent functioning cells, each ready to begin the cycle again. Chromosomes are the structures that carry genetic information in the form of DNA. During mitosis, it's vital that chromosomes are accurately duplicated and distributed to ensure each daughter cell receives the correct genetic instructions. Errors in this process can lead to genetic disorders or cell malfunction, highlighting the importance of precise chromosome handling. Cell division is a dynamic and highly regulated process. Cells constantly monitor their environment and internal state, deciding when to divide based on signals and needs. This dynamic nature allows organisms to adapt, heal, and grow in response to changing conditions. Observing cell division under a microscope reveals the incredible choreography of life at the cellular level. While the basic steps of mitosis are similar in plants and animals, there are key differences, especially during cytokinesis. Plant cells form a cell plate, while animal cells pinch in with a cleavage furrow. These adaptations reflect the structural differences between plant and animal cells, ensuring successful division in both kingdoms. Sometimes, errors occur during cell division, leading to problems like cancer or genetic disorders. Cells have built-in checkpoints to detect and correct mistakes, but when these fail, the consequences can be serious. Understanding mitosis helps scientists develop treatments for diseases caused by faulty cell division. Growth in multicellular organisms depends on cell division. As cells divide, tissues and organs expand, allowing the organism to grow larger and more complex. This process is especially rapid during development but continues throughout life as cells replace those that are lost or damaged. We've journeyed through the remarkable process of mitosis, from interphase to cytokinesis, and explored how cells divide to enable growth, repair, and reproduction. Understanding mitosis gives us insight into the continuity of life and the incredible complexity of living organisms. Whether in plants or animals, cell division is the foundation of life. Thank you for joining us on this exploration of the cell cycle and mitosis.